This part of it is on the incus, but then instead of the rest of the block, it has a handle. What you do with the anvil is you hold the metal against it with some tongs, and you beat on it with a hammer. The head of the malleus is touching the incus. So it looks a little like an anvil, and you hit it with a hammer, sort of. Stapes is stirrup. This is the one that really looks the most like what it's called. The part that would hang from the saddle is against the handle of the incus. And then where your foot goes is against the inner ear. The ossicles together transmit the vibrations from the tympanic membrane to the inner ear. And the last structure of the middle ear is, there are a couple of different names for it. Auditory tube, eustachian tube, and the one that tells you the most about it, but is probably the hardest to remember, pharyngotympanic tube. It goes from the tympanic cavity to the pharynx. So when your ears pop, that is air moving through the auditory tube, either in or out. That's there to equalize the pressure between the tympanic cavity and the air around you. So it's usually collapsed, but when you open your mouth, like with chewing or yawning, that can pop it open and the air can go through. If the pressure is unequal, then one side will be pushing harder against the tympanic membrane and it won't vibrate as well. That's why everything gets louder after your ears pop. Any questions on the middle ear? On our models, you can see the auditory tube here. The malleus and incus are on the piece with the tympanic membrane. The incus, the pointed one, malleus attached to the membrane. Tympanic cavity here. And the stapes is attached to the inner ear piece. And I think some of these are broken. So if you don't see a little wishbone looking thing on there, you've got one of the broken ones. But we will have all of these. Yes?
My daughter has tubes in her ears. Does that make sound louder? Or?
It can. The tubes, I had this done when I was a kid, they are placed in the tympanic membrane. And so that lets a little bit of air go through so that the pressure can equalize all the time. So if you have an ear infection, there's fluid in the middle ear, and so often they put the tubes in to help drain that as well as to equalize the pressure.
I just never asked her doctor anything if it makes anything louder or anything for her. It doesn't seem to affect her, but
yeah, it might have, depending on how her hearing was before they put them. She might have it back. Because when I got it done, it was constantly, my parents would say something and I'd go, what? And at first they thought I wasn't paying attention. I was focused on my book or whatever else I was doing. And then one day my dad says, you know, maybe she actually can't hear us. And
she had really bad balance issues and ear infections from like the day she was born to 8 months. So
yeah. Yeah, and so infections can, basically you get things inflamed in here. There's a mucous membrane in the tympanic cavity. That can interfere with the vibrations. It can put fluid, which changes the pressure, all kinds of issues. The inner ear, I mentioned here, encased in the temporal bone, but so is the middle ear. Another word for the inner ear is the labyrinth. Anyone know what a labyrinth is? Yeah, it's like a maze. term was first used in Greek mythology for the big complicated area you have to get to, and it's guarded by the Minotaur. And then there was that awesome 80s movie with David Bowie, where she had to get through the maze to get her little brother back. So there are multiple channels and chambers inside the inner ear. That's why they call it the labyrinth. From the outside, what you see is one bulge, which is the vestibule, a spiral, which is the cochlea. Cochlea actually means snail. And then 3 tubes that are connected to each other. These can be a little tricky to draw flat because of the way they overlap. Those are the semicircular canals. Now the border between the vestibule and the cochlea isn't always as obvious as I drew it. On here you can kind of tell because there's a thicker bulge and then it gets thinner, and that's where it's the cochlea. The stapes actually connects to the vestibule. So the vibrations are going to pass through there, but the vestibule itself doesn't have any hearing receptors. Those are all in the cochlea. On here, some of these have cut inside the cochlea, and they've got colored channels there, and that's what we'll see here. And the vestibule is the bulge and semicircular canals, and they are filled with liquid. They did that with the models, but it's dried out over time, so it doesn't flow the way it normally would. Now, in the cochlea, you cut across anywhere along the spiral, you'll find these 3 chambers. The center channel is called the cochlear duct, and it's the one that has the hearing receptors in it. In it is a fluid called endolymph. The outer 2 are both scala something. You'll see it in the lab book. You don't have to remember it, but go ahead and fill it in. They are filled with perilymph. The fluid never connects from one to the other. There's endolymph and perilymph in other parts of the ear as well. So if we follow the sound vibrations, they go through the external acoustic meatus, they vibrate the tympanic membrane and the ossicles in the order they're usually listed, malleus, incus, stapes. Then they vibrate the perilymph from the vestibule to the cochlea, and the vibrations transfer from the perilymph to the endolymph, where they'll be detected in the cochlear ducts. This is called the spiral organ, or the old term for it is organ of Corti. So, spiral because it's in the spiral of the cochlea. So this is a section of spiral organ isolated from the cochlear ducts. The receptors are these little blue guys shaped sort of like champagne flutes. They're called hair cells because they have little hair-like things sticking up from them. They're on kind of this hill of tissue, and if you can see these yellow lines here, it's where they connect to the neurons. Now, this is the membrane dividing the cochlear duct from the lower scala. The little hairs of the hair cells are reaching toward this immobile plate which I probably should have put the name on here. It's called the tectorial membrane. You'll see that in the lab book too. The way hearing works is the vibrations shake the membrane between the two chambers and make the hair cells bump up and down. They run into the tectorial membrane and bend, and that causes the hair cell to stimulate the neuron. Different groups of hair cells along the length of the cochlear duct respond to different frequencies of sound, and that's how you can hear multiple notes at the same time. From the spiral organ, the messages are passed through the vestibulocochlear nerve, cranial nerve number 8. This name tells us that it's collecting from the cochlea and the vestibule, which we'll see coming up. The messages go to the medulla oblongata, then the midbrain, and then the thalamus. So they go into the midbrain so it can process any necessary reflexes. And then from the thalamus out to the auditory cortex. Which lobe was that in? Temporal. Yep. And out. Questions on hearing? Before I do equilibrium, if anybody wanted to go to the schizophrenia talk, that is over in JCCB in 10 minutes. You are excused. I didn't sign us all up because it was important that we finish the chapter today. But if you want to catch that, I will have the recordings posted and you'll be able to catch up that way. They might not have lunch for you if you didn't sign up, but there's plenty of room. Equilibrium. This is picked up in the vestibule and semicircular canals. Together they're called the vestibular complex. There are two components to it, called static equilibrium and dynamic equilibrium. Both of them send messages by the vestibulocochlear nerve. So it'll have connections to each portion of the semicircular canals and the vestibule, and those come together and they join together with one from the cochlea. Static equilibrium, static like homeostasis, standing still. Basically it detects gravity, but it's also affected by inertia. The vestibule has two chambers called the utricle and saccule that work together to detect which way the strongest force is pulling on your head. Most of the time that's gravity, and so it'll tell you if your head is upright or tilted or lying down. Inertia can throw it off. Think about, if you speed up really fast and you're pushed against the seat, that's stronger than the gravity and pulls on the sensors in the vestibule. And there's that carnival ride where it spins around and then it goes up but sideways, but you still feel like you're lying, your back is down even when you're at the top of it and your back is up. That is inertia overriding gravity, and that'll affect that both holds you against the wall and affects your sense of static equilibrium. In both the utricle and the saccule, there's a structure called a macula that has hair cells. In the hair cells are otoliths, which means ear stones. So the gravity or inertia pulls on them and bends the hair cells. It looks something like this. Here's our hair cells, and here's another case where they're actually neurons. The only place I really want you to remember that is for smell, but you might be noticing it here. So there's this gel with the otoliths in it, and when the angle allows, the otoliths pull down, pull the gel, and bend the hair cells. Questions on static equilibrium? Dynamic equilibrium. Dynamic means changing. This detects movement and rotation. It's picked up by these semicircular canals. Each one is filled with perilymph and has a sensor at one end called an ampulla. In the ampulla is a cupula, a little mound of gel with hair cells in it. So all three sensors in the ear have hair cells, but they all look a little different and work a little differently. When the perilymph shifts, it moves the gel of the cupula, and that bends the hairs of the hair cells. So here are the hair cells in blue attached to neurons. They have longer hairs that go out into the cupula. So with movements, the cupula bends as the perilymph moves.